The natural killer cells and macrophages activate each other at the site of infection. Okay? Who is going to invite natural killer cell to the site of infection? This is the macrophage. Okay? The macrophage has to invite the natural killer cell to the site of infection. I have told you that macrophages, they are the principal cells that are going to, tackle the pathogen and to inform all other cells to come to the site of infection and deal with that pathogen. Okay? Now, an activated macrophage, it secretes interleukin 12. This is a cytokine, and it used to be known as natural killer cell stimulatory factor. People used to call it natural killer cell stimulatory factor. So in the tissues, there are tissue resident natural killer cells. Some of the natural killer cells have to arrive from the blood, okay? But in the tissues, there are natural killer cells that are resident in tissues. In tissue resident natural killer cells, not yet activated by the interferon response, okay? Interferon has already been produced as a result of infection with viruses, But some of the natural killer cells that have not been activated, they have not come in contact with those interferons produced, as you saw in the previous slide. They are activated by this cytokine interleukin 12. Where is it coming from? The macrophage. The macrophage has come in contact with the virus. It has detected the virus. The macrophage has secreted interleukin 12. Natural killer cell has receptors for interleukin 12. So this cytokine would bind to its receptor. So this is a signal for the natural killer cell's activation. Okay? And the cells that are going to be recruited from the blood are going to join these cells with the help of a chemokine CXCL8, the gradient which is formed by the macrophage for inviting different cells to the site of infection. So in the previous lecture, you saw that neutrophils were traveling along that gradient to the site of infection. The natural killer cells also are going to travel along the same gradient because they also have receptors for CXCL8, the chemokine factor secreted by the macrophage. Okay? So this macrophage is not only activating the natural killer cell which is present in the tissues, this natural macrophage is also inviting the CD56 DIM natural killer cells that are there in the blood vessels to please come here to the site of infection. Okay? So interleukin 12, what is it doing? It's activating the natural killer cell. The natural killer cell macrophage conjugate, the synapse delivers interleukin 15, which with interleukin 12 activates the natural killer cells. Interleukin 15 also is playing a role here. Both interleukin 12 and 15, they help with proliferation and differentiation of the natural killer cells. And this delivery is happening right here when the natural killer cell and the macrophage, they would come closer to each other and make this synapse. The synapse is a combination of receptors that are bound to each other. Okay? The receptors would just, concentrate in this synapse. And I had told you that changes would take place both in the macrophage and the natural killer cells. The microtubules or the microfilaments would organize themselves in such a way that the cargo would be directed towards the synapse. The cargo can either be cytokines, or in some cases when the synapse is made with the target cell to be killed, then the cargo can be these granules which are filled with different enzymes like granzymes or perforins or other glycoproteins. Now what happens next? The natural killer cells proliferate and differentiate into effector natural killer cells secreting interferon gamma. Okay? And effector cells, natural killer cells, the natural killer cell which has become an effector cells due to the secretion or coming in contact with interleukin 12 and 15 is going to secrete interferon gamma. These effector cells are not the cells that are going to kill other cells. The natural killer cells here is not the CD56 dim cytotoxic, rather it is CD56 bright cell activated, which now is going to secrete this cytokine interferon gamma. Okay? Interferon gamma is the principal cytokine produced by natural killer cells in innate immune response. We call it type 2 interferon. The type 1 interferons are interferon alpha and interferon beta. Type 2 interferon is interferon gamma. So this is the principal cytokine produced by the natural killer cells in innate immune response. And the principal target of this interferon gamma in innate immune response are the macrophages. The interferon gamma secreted by this effector natural killer cell is going to act back on the macrophage now. Okay? Here the macrophage is helping with proliferation and differentiation of the natural killer cells. The macrophage is secreting interleukin 12, which is helping with differentiation and proliferation of the natural killer cells, so the natural killer cell will become effector cells. And these effector cells are secreting interferon gamma, which acts back upon the macrophages and help with several functions of the macrophages to be improved. For instance, increased or improved phagocytosis. More cytokine productions, more production of molecules that would lead to elimination of the pathogens. It means that the macrophages' capability to eliminate the infection would be increased. And that would be increased if the macrophage is more eager or more hungry, and it is going to engulf on more of the viruses that are present in the tissues. If it is secreting more cytokines like interleukin 12, CXCL8, tumor necrosis factor alpha, the pro-inflammatory cytokines, in order to invite other cells like the neutrophils and these natural killer cells and other cells to come here, dendritic cells, to come here and deal with the pathogen or neutralize the pathogen or engulf the pathogen. Okay? So you see that this is a positive feedback loop between a macrophage and a natural killer cell. They are just helping activate each other, and in this way help with elimination of the pathogen. Now, the natural killer cell also has something to do with dendritic cells. You know about the dendritic cells. These are the myeloid dendritic cells that we would talk about now. Previously, I had told you about plasmacytoid dendritic cells, if you remember. Now, the myeloid dendritic cells, the natural killer cells, you know about its function. What is the job of the myeloid dendritic cells, or simply dendritic cells? What do these cells actually do? Do you remember? Dendritic cells are present in the tissues, just like the macrophages. They have dendrites, that's why they are called dendritic cells, and their job is to sample antigens from the environment. Okay? So they can sample different antigens in the tissues. When there is entry of some pathogens like viruses or bacteria or some other microbes, then these cells would just capture them and figure out what kind of antigen is this and what kind of response is needed against them. So activation, proliferation, and differentiation of the natural killer cells are driven by the dendritic cells. Okay? When these natural killer cells, they come in contact, for instance, with a dendritic cell, okay, the dendritic cells can either, sample antigens in different, tissues, just like in intestines. I think we have not gone through that stuff. We have skipped that, but they are very important in intestines. The dendrites come out of the luminal epithelium, and they sample antigen from the lumen of the intestines as well. In intestinal mucosa, there are lots of these dendritic cells that are helping with the adaptive immune response. They are sampling antigens, presenting them to the T cells, and the T cells then become effector cells, and these follicles that are present there in the intestines. Similar is the case with other tissues, but as you know that majority of these pathogens enter your intestines or your oral cavity, gastrointestinal tract is always being challenged by many different types of pathogens because you are eating different things, okay, which can contain these different types of microbes. That's why the natural killer cells are very active there. Okay. Now what happens that the interaction of the natural killer cells with dendritic cells can have several outcomes. In a virus-infected tissue, an immature dendritic cell expressing viral antigens can activate natural killer cells and drive their differentiation into effector cells, the left panel that they are talking about. When activated, cytotoxic natural killer cells are abundant, as you see in the middle panel. This is when it would secrete interleukin 15, and the interleukin 15 again would act upon the natural killer cell because it has got receptor for interleukin 15, and interleukin 15, just like interleukin 12, you saw in the previous slide that both of these cytokines are required for proliferation and differentiation of the natural killer cells. So when this NK cell is now in contact with the dendritic cell, which was doing sampling of antigens in the tissues, this natural killer cell now has come in under the influence of interleukin 15 secreted by the dendritic cell, and as a result the dendritic cell would proliferate and differentiate to become an effector cell. Okay? Now upon this contact you see that proliferation and differentiation of the same natural killer cell has been caused by interleukin 15 secreted by the dendritic cell. Now there are two things that can happen. When natural killer cells are abundant, they outnumber dendritic cells and kill the dendritic cells. Okay? If this proliferation is so great that lots of these natural killer cells, effector natural killer cells are produced, then these natural killer cells can kill the dendritic cells that help it with proliferation and differentiation. Okay? This is the same dendritic cell which secreted this molecule to help with proliferation of this cell. So they would grow in number and they would become effector cells. So if they just outnumber the dendritic cells in the tissues, these natural killer cells are going to kill this particular cell. For doing that good to the natural killer cells here to help with its proliferation. Okay? Another thing that can happen is that if the natural killer cells are not able to deal with infection, or they could not eliminate the pathogen from the tissues, then the natural killer cells, the dendritic cells, they would outnumber the natural killer cells. There are no enough natural killer cells to deal with all the pathogens in the surrounding or in the tissues. There are more dendritic cells now in the tissues. The natural killer cells, that stock has been exhausted. Now what would happen in this case? Natural killer cells are scarce. They signal dendritic cells to mature and initiate an adaptive immune response. Okay? Now, what would happen? The innate immune response is not successful. The natural killer cells cannot cover the infection. They cannot kill all the pathogens. So this would result in the production of motile natural killer cells that would move from the tissues into the lymphatics. They would enter into the lymph nodes and carry these antigens along with them to be presented to the T cells, so that the T cells and B cells would mature there in the lymph nodes. Okay? There would be B cells and there would be T cells with specific receptors, and maybe the antigens that they are taking inside, these viral antigens, would fit somewhere with some kind of B and T cells in those cells would stay there in the lymphatic system. They would proliferate, and that adaptive immune response that we had talked about, that response would be initiated. Okay? So the second scenario, where the natural killer cells are not very active, or there are not many natural killer cells in the tissues to eliminate pathogens, this would help dendritic cells to modify themselves, become motile dendritic cells, and take this antigen very quickly to the site of to the lymphatic system, because there is a need. This is an emergency. Somebody should take this message very quickly from the tissues into the lymphatic system to activate another type of immune system against them, which is the adaptive immune system.